Section 8.5, Translation. Translation converts the messenger RNA messages that were made during transcription in the nucleus into amino acid chains, or polypeptides. One or more polypeptides will make up a protein. 4 nucleotides, adenine, guanine, cytosine, and uracil, are used to code for 20 different amino acids. DNA and RNA can be separated into 3-letter segments. These 3-letter segments are called codons. Okay, and a sequence of 3 nucleotides that codes for a specific amino acid. An amino acid can be coded for by more than one codon. We'll talk more about that later. In addition to coding for amino acids, one codon will code for the start codon. The start codon is AUG, adenine, uracil, guanine, and this codes for the amino acid methionine. Okay, so translation always begins with methionine. This methionine is later going to get taken off during the protein building process, the final tweaking of the shape, the twisting and formation of hydrogen bonds to get its final shape. The methionine will get knocked off. There are also 3 other codons that code for stop codons, which signal the end of translation. So a stop codon does exactly what it says it is. It stops translating the messenger RNA. The genetic code matches each codon on the messenger RNA to its specific amino acid that it codes for, or the function. Now remember, these functions would be either start or stop. Remember, there's 1 start codon and there are 3 stop codons. Codons are always read without spaces as a series of 3 non-overlapping nucleotides, which is called the reading frame. Any changes can change the resulting protein. For example, we have this sequence of messenger RNA, and I just put lines between each 3 so it's easy to see where these codons are. So these 3 codons, CGA, codes for arginine. These next 3, UAC, code for the amino acid tyrosine. The next 3, AGU, code for serine, and AGC also codes for serine. If this reading frame shifts 1 unit to the right, so now it's the same exact sequence, but the reading frame shifts over 1, now we're getting GAU, which codes for a different amino acid altogether called aspartic acid. ACA codes for threonine, and GUA codes for valine. So by just moving this 1 base, this reading frame moving over 1 base, it changes the entire sequence of amino acids after that point. Regardless of the organism, codons code for the same amino acid. Because this codes for the same amino acid, it's known as the universal code. This gives evidence that almost all organisms must have come from a common ancestor. Okay, this is a picture of the genetic code, which is also found on page 472 of your book. Okay, 472 of your iBook. Okay, so let's just see what you get so far. Suppose the messenger RNA molecule in the cytoplasm had 300 nucleotides. So how many nucleotides per codon? So there's 3 nucleotides per codon. So if there's 300 nucleotides, that means there are 100 codons. So if there's 100 codons, does that give us 100 amino acids? Well, the first one would be start, and the last one would be stop. And so it would be 100 minus 1 minus another one. There would be 98 in the protein. Okay, so use your book on page 472 to follow along how to do this. So we have a DNA sequence of TAC, TAA, ACA, CCG, and ACT. So during transcription, the messenger RNA sequence would be AUG coming from here, and it would be CUU from here, UGU from here, GGC from this guy, and UGA. So those would be the codons on the messenger RNA sequence. Now looking on page 472 of your iBook, if you look on that, the first base, which would be A, so you're going to look on the left side of this, A, and you would find a column labeled A, the row labeled A, and then UG would give us methionine, which is the start codon. Okay, so that's for all the info. The second one, CUU, so again, you start on the left side where it says C, that's the first base, and then go to the middle, where's the U column? The U column is the first one in red, so CUU would give us leucine. Okay, UGU, first base is U, the second base is G, and the last base is U. That gives us cysteine. Next, the messenger RNA sequence is GGC. So we go to GGC, which gives us glycine. And last, we have UGA, and UGA gives us a stop codon. Okay, so you get all this stuff from that messenger RNA genetic code for messenger RNA codons on page 472 of your book. Okay, so ribosomes are organelles in the cytoplasm, and remember they're either in the cytoplasm floating around freely, or they're on the rough endoplasmic reticulum. Okay, and these ribosomes are the sites of protein synthesis. Site means where it happens. Okay, these are where the amino acids are bonded together. Ribosomes consist of two subunits. The big subunit, which would be this, has three binding sites for tRNA, or transfer RNA. Remember, that's one of the three types of RNA. This part of the large subunit is called rRNA, or ribosomal RNA, because it makes up most of the ribosome. The small subunit binds to messenger RNA. So this small subunit is where messenger RNA is going to bind so it can get read and help create a protein. So tRNA acts like an adapter between the messenger RNA and the amino acids. Okay, so looking at our tRNA molecule, we have amino acids on this end, and on this end we have something called an anticodon. The anticodon is a set of three nucleotide bases, that are complementary to the messenger RNA codon. Okay, so this anticodon is part of the transfer RNA molecule. So if the messenger RNA sequence was GGG, CAU, UUA, and CCG, the tRNA sequence, which would be the RNA complement to GGG, would be CCC. The complement to CAU would be GUA. From UUA, it would be AAU. U, and from CCG, it will be GGC. So these would be the anticodons found in the transfer RNA that would link up or bond to the codons that would be on messenger RNA during translation. Okay, so these translation animations, this is just something to kind of show you a different idea or a different way of looking at this whole process of translation. You may have to copy paste this into your browser. I don't think it's working right just to click on it and have it come up. So you may have to copy that into your browser or type it into your browser and hit it. It's pretty short, it's about 15-20 seconds, but it gives you a good visualization of translation. Okay, so a quick overview of translation. First, step 1, ribosome binds to messenger RNA. The tRNA binds to a start codon on messenger RNA, and remember the start codon only codes for one thing. It codes for the amino acid methionine. Methionine is always the first amino acid in the chain of amino acids that's making up a protein. Step 2, the ribosome pulls the messenger RNA strand the length of one codon. And what forms are polypeptide bonds between the amino acids. So if we have, this is our first codon on the messenger RNA, which binds the anticodon, which codes for methionine right here. This next tRNA molecule will link up with the 3 bases right next to the first codon. Okay, so link up and bring in this amino acid leucine. The peptide bond forms between methionine here and leucine here, and by these tRNA holding them in close proximity, it allows for them to bind up, forming this peptide bond, a specific type of covalent bond that holds amino acids together in protein formation. As the bonds form, the empty tRNA molecule exits the ribosome. Once it gives up its amino acid, it will unattach from the messenger RNA and go look for another amino acid in the cytoplasm that would be coded for by its anticodon. Okay, when a stop codon is reached, the ribosome releases the protein, and protein synthesis is done. Okay, again, this is just that same animation. Again, try it, look at it. It's pretty helpful to get you to visualize what's going on with this process of translation.